cognitively healthy. So we could not assess that. In a paper, you cannot find the clinical penetrance, we could not assess it. But of course, because there is a lot of research in this field, in literature, you can see that it's established, and this is, for instance, a Nature Medicine paper of this year, that the lifetime risk of AD dementia is probably in the 40 to 60%. In this study, it was 60%. The lifetime risk of developing dementia if you are APOE4 homozygote. Let me introduce you the nuance that if you add MCI, that all agree is symptomatic Alzheimer's disease, this number will be even higher. Of course, if you add pre-symptomatic AD and then you move away from the international working group to Alzheimer's disease, the number would be even higher. But even with the symptomatic stages, again, rules, the lifetime risk of APOE homozygotes is at the same level as many diseases outside neurology. And what happens in neurology? In neurology, there are diseases that no one doubts are of genetic origin. For instance, the C9 expansion. And here there is a controversy of what's the clinical penetrance of this mutation. But the estimates in some papers, recent papers you can see here, are less than 40%. Why is it a mutation that confers less than 40% risk for the clinical syndrome? Genetic in other areas, and something that confers a 40 to 60% risk is not genetic because it's not fully penetrant. That's a thing that, again, we really need rules and be consistent, even across neurology and across medicine, on how we define the risk conferred by a mutation. The racial thing is even more important. And because there is very good epidemiological evidence that the risk conferred by the APOE gene, haplotype, zygotes and heterozygotes, here, homozygotes to the left, heterozygotes to the right, and here you can see East Asian, non-white, non-Hispanic white, non-Hispanic black, and Hispanic. And you can see that the odds ratio conferred by the haplotype clearly is different across the different ethnic groups, such as that East Asians are considered to have the highest risk if they are carriers of the APOE4, and Hispanic and non-Hispanic black are considered the risk conferred by the APOE haplotype is considered to be smaller. I have to say, however, that all these studies have been done by clinical genetic studies. So here, most of what we know in this kind of studies is done by a clinical diagnosis of the disease, oftentimes or most often not supported with biomarkers. And that can be one of the reasons why we are seeing such important racial difference. But in any case, this is hugely important because until we really solve these issues, it's very difficult to really inform and do the genetic counseling to a homozygote because it's very difficult to know what is the specific risk for that person, because there is no doubt, and I would agree with the people that are concerned about the implication, that what a person is really concerned of is not whether he or she will develop amyloidosis when they are 80. What they are concerned is whether or not they will lose memory of whatnot, even more cognitive complaints. So we really need to estimate these risks and really understand the effects of the ethnic background. So in order to do that, we team up with Claudia Suemoto in Sao Paulo. And the reason to do that is because in the NAC database, the one we analyzed, basically there is not enough ethnic diversity. You go there, almost all the brains that are available in the NAC database are white. So here in Sao Paulo, they have, which is a huge city in Brazil, as you all know, they have many deaths, as in a big city, but they have a very interesting autopsy service that in those people that do not have a clear diagnosis of non-traumatic deaths without medical assistance or undetermined causes during life, they'll do a lot of autopsy. So it's a very clear source of having many brains analyzed with there. Of course, they're healthier and probably they have less dementia than what it's a different bias. It's very difficult to escape bias when you're doing pathological studies, but it is population-based and it has a lot more diversity. And they are collecting many, many brains and doing an amazing work there in Brazil. So they've collected over 2,600 participants. So our paper in with the whole of the US, it was based on in the end up ended up with 3,000. So just imagine how huge effort they are doing there. They have much more diversity there, and this is, as I said, it's a bias towards much more asymptomatic individuals. So it's very different, a very, very different picture from the NAC database in the sense that it's much more ethnic diversity and many people that died with no symptoms whatsoever of the disease, whereas in the NAC database, it was symptomatic individuals. So that's very important to consider the disease. So here what we analyzed is that here the biological penetrance of the disease, so the BRCA stages, yes, you can see the different haplotypes, so 4/4s, 3/4s, 3/3s, 2 something. So if you look at the left column, you can see that, of course, the more gene dose you have, the more red, yellow that you have. There's no doubt that also in this data set, also in this cognitively unimpaired, diverse population, the impact of ApoE4 is huge. It might not be fully penetrant, could be. Do bear in mind that this is biased to more cognitively impaired, but there is maybe the penetrance is lesser than in the bias samples that we had in the Nature Medicine paper. That's an important result. But another very interesting result is that here, when you analyze the biology, when you analyze the pathology in this case of the disease, we did not see that Black participants had less AD than the white participants. So this clinical genetic association that clearly have been established in the literature, we could not see in this study. So let me end up for a call for action to prioritize APOE homozygotes in prevention and therapeutic trials. I think that this is extremely important, and there are reasons to be very optimistic, because I do believe we can defeat AD for homozygotes and the genetic population. I do believe we can do it, and we really need to tackle this problem. So this is something that is reasons for optimism. I think that now, if you're an APOE4 homozygotes, you should read this slide. And clearly there are reasons for, of course, we need the data. We need a positive trials to prevent the disease. But the same drugs that we are now administering in symptomatic participants, I'm talking about Leqembi and Kinsula, the donanemab and lecanemab. Now they are being tried in the HEAD study, lecanemab, and Lilly is also having secondary prevention trials. The HEAD study is fully enrolled. It's going to read out in 2 years, and it's going to tell us if removing amyloid in the presymptomatic stages of the disease delays the onset of symptoms. And if this happens, it will be completely different scenario. We'll have the answer in just 2 years. And it's just the spearhead of many other trials that are coming in the future. But we do have some indirect evidence. This was a paper that was published earlier this year in the Lancet Neurology by the group of Randy Bateman in WashU in the Diane study. And there they took a trial that had been negative, gantenerumab. You know that gantenerumab is a trial that did not remove amyloid as efficiently as lecanemab or donanemab, but removed amyloid to a certain extent. And so basically here they followed individuals for many, many years. And there they have, with small numbers at the end of the study, but those that had received gantenerumab for longer, they had 50%, more than 50% reduction of progression to symptomatic AD. Again, this is a failed trial and very long and very small numbers at the end of the study, but evidence to suggest, in agreement with the rationale, that maybe administering and removing amyloid, at least in these genetic populations, can delay the onset of Alzheimer's disease, the symptomatic Alzheimer's disease, significantly. So, of course, there are more trials in autosomal dominant AD, trying newer generation of these drugs. hopefully we'll start doing the same, or we're already doing the same in in Down syndrome with active vaccination, and hopefully soon with antibodies too. So I think there is reasons to believe that maybe in a few years we might have drugs that are effective in delaying or preventing Alzheimer's. So reasons for optimism. So in order to do that, we need to have trials in ApoE, focused on ApoE homozygotes. And that's not only because if you move preclinical and certainly to primary prevention, for instance, the numbers are much smaller because homozygotes represent around 15% of symptomatic AD patients, but only 2% of the general population. So we really need to enrich for homozygotes. And that's because there are biological differences. So for instance, the response to and this is a problem, I'll show you some things that are more optimistic, but the side effects of the drugs that we have for Alzheimer's disease have a huge gene dose response to. So if you are a homozygote, you do have more risk of developing side effects if you are receiving these drugs. As you can see here, for instance, here is area H, but with area E, edema and these hemorrhages, the risk for developing some side effects is much higher if you are homozygote. So clearly, if you have a differential response, be it risk, be it efficacy, we really need to analyze the data separately. Luckily, there have been some people already in the field that have recognized that for much earlier than we did. And so, for instance, Alceon, they ran the Apollo E4, and this is really interesting for many things. Basically, because in this and the next example, they are doing repurposing of drugs. So here, the tramiprosate is a trial that was negative, not a failed trial, a trial that was negative a few years ago in Alzheimer's, but they clearly had the patience and the intelligence to look into the data and think that maybe ApoE4 homozygotes would benefit for tramiprosate, which is a molecule that inhibits the oligomerization of A beta oligomers. And although they recently published the main results and didn't find that significant overall cognitive benefit, in a pre-specified analysis, they saw that maybe patients in the MCA stage of AD, they might benefit here. And they had a very favorable safety profile, and it was an oral compound. And they also showed some very encouraging data in the rate of brain atrophy. So again, this is repurposing and recognizing that ApoE homozygotes might have a different perspective. A different trial that is now ongoing, and is probably Dr. Ashford can tell us much more about it, is we met at night, which is something that we are doing for hypertension, basically in medicine. But Dr. Wang clearly identified that very specific transcriptomic reversal of ApoE specific signature. That means that he recognized that ApoE4 was having a very specific signature, and analyzed what compounds that we already were using in medicine could reverse some of these studies. They went on to animal models, and they showed that drug could prevent some of the hallmarks of the disease in the animal models. They went to databases of people that had been using this drug in the general population and showed that maybe that these people showed less AD-DMS. And then they move on to a clinical trial run at Stanford to really see if this worked. So again, it was the intelligence of focusing and analyzing the distinctiveness of ApoE homozygotes and trying to in this case, take advantage of the knowledge that we gain to really fight Alzheimer's in this subpopulation of the disease. And this is another very recent example of published this year also in Nature Medicine in July. And here what they did is analyze another huge database, mainly composed of women, but then a replication cohort of men that follow individuals for many years, 1989, sorry, and this is 30 years more than 30 years of follow-up. So in Alzheimer's, we need long follow-ups, and we really need large numbers. This is 6,000 individuals. What did this show us? Again, in this study, if you are a homozygote, the risk in this study for these many months, you can see it's over 40%. And this is a longitudinal prospective study. And probably if we increase the follow-up, probably the numbers increase. So again, confirming this ultra high risk or very high risk of symptoms onset. Again, this is for dementia. If we include MCI, it would be higher. But the very interesting thing is that they found that the metabolic profile on ApoE4 homozygote was different from that of heterozygotes and non-carriers. And what's really nice is that the Mediterranean diet seemed to be much more effective on homozygotes than non-carriers or heterozygotes. So the people that benefited most from a healthy diet were those that were homozygotes. So again, we really need, if we want to do precision medicine, probably the best place to start is by analyzing the ApoE genotype. Because I think that together we can defeat Alzheimer's and Down syndrome. And here is time for action. Really, what you are doing, just gathering together, lobbying, and contributing to studies, this is the way forward to fight the disease. We really need to clarify the ApoE biology. We really need to define more cells type-specific mechanisms. We really need, I do believe, construct ApoE4 enriched cohorts. We really need to understand better the biological versus clinical risk across disease stages in diverse populations, so we truly can inform and do the genetic counseling to the person with really good data. And we need to identify the best therapeutic timing for the different drugs at the different timing of the disease. And certainly we need targeted ApoE-directed therapies, because we did not consider it causal, because we did not consider it a genetic form. Most of our attention has gone to the mechanism identified in autosomal dominant AD, in presenting 1, 2, and APP mutations. We certainly need to do the same kind of investment, if not more, in ApoE-related therapies. And this is, for instance, something that we hope we'll be able to do in Barcelona in the next few years. We're trying to collect and raise funding for the BRAVE AD trial. It has been mentioned, ApoE4 is not just a question only of the brain. Of course, it's a systemic. It also has vascular components. It might affect the eye. And so this is a cohort that we want to launch, really enrich for ApoE4 homozygotes. So basically we'll recruit several hundreds of people with homozygotes. We'll also include some people with ApoE2, that I think is another very interesting population. And we're going to try to do multimodal assessments, build a cohort there and try to nest clinical trials and accelerate the disease in the coming years. And with that, I hope I've convinced you to provide evidence of why not only do we have an elephant in the room with Alzheimer's, and with all my respect, we do have a mammoth in the room, which is ApoE4 homozygote. As I said, this would mean that 15 to 20% of the symptomatic AD would be genetically determined. And this is my true belief. All my research in the last 20 years has revolved about these genetic forms of Alzheimer's disease. As I started with AD, and then I moved to Down syndrome associated Alzheimer's disease, and now we're putting all of our efforts trying to fight Alzheimer's in ApoE homozygotes. And with that, I'll finish and saying that ApoE4 homozygotes, in our view, should be included in the growing family of genetically determined AD with near full biological penetrance. Because of the predictability of this biological penetrance, the predictability of disease onset, the sequence of biomarker changes. And because of this predictability, these populations are ideal to perform secondary prevention trials. And really, we need to analyze the differential response to treatments, both in terms of efficacy and in terms of safety. And all the trials should be powered to really analyze if they are equally effective and safe in ApoE homozygotes. And as I said, that this is my true belief that AD, because we know the cause, might be easier to cure in this population than in the general population, in which probably a myriad of environmental factors are playing a larger role and might be more difficult to identify subpopulation. Here we have more homogeneity, and this should help us fight the disease. And with that, I'll end up by thanking all the participants. I always put people with Down syndrome. They are a vulnerable population that really teaching us a lot. And I think that the work that we did in EPOI homozygotes in depth a lot to the research we did in them, but of course to all the EPOI homozygotes that have participated in our studies, all our colleagues, and the memory unit in Barcelona. And with that, I'll be very happy to take questions.